So let's get this going. Again, the nature of the slides, they're a little bit different structured from what's on Canvas. It's really just an order. There's no new material. I wanted to be able to present the figures first and talk a little bit about what we see and then summarize the answer to this question. So our first question that we're going to deal with is this one. What are the stages of the cell cycle and how do chromosomes factor in? What's going on with the chromosomes at any point in the cell cycle? What I want to encourage you to do is this. Whenever you're thinking about the DNA of a cell, or you're thinking about anything the cell does, always think about it in the context of the cell cycle, where the cell's at. Most cells, at any given point, if they are doing what that cell is designed to do, like if they are a neuron, or they are a liver cell, or they are a cell lining your intestinal tract, they are almost always in G1. Now they may be in a place where they've completely left the cell cycle. It's still G1. We just don't call it G1 because G1 gives you the idea that S is coming next. But if S never comes next, then it can't really be in G1, can it? It's completely left the cell cycle altogether, and we call it G0 or G0, but really it's just G1. It's just there's no S coming next. But I want you to always think about these in context of the cell cycle. And so during G1, you can think of this as the beginning of the cell cycle. There's really no real beginning. There's no end. It's a circle, right? And that's the nature of a circle. There's no beginning and there's no end. It just depends on where you want to start and then go around a full circle, right? So if you start at 12 o'clock and go around a full circle, that's one thing. But you can very easily start at 3 o'clock and go around a full circle. It doesn't really matter. There's no real beginning. But G1, G stands for growth or gap. Different things. Gaps the more frequently used. And so when you look at this in the cell cycle, according to this diagram, what is the longest part of the cell cycle? S, right? You're looking at this and S is just really, really wide. Now, why is that interesting? Anyone think about why that might be interesting?
Does it take a long time to replicate DNA? And then G1?
It actually doesn't, right? We talked about this. And DNA polymerase, each DNA polymerase can write 1,000 bases a second. And you're like, okay, well, our genome's 3 billion bases. So if you're only doing 1,000 a second, that's 1 million seconds, actually 3 million seconds to replicate the genome. Well, yeah, that's if it was being done by a single DNA polymerase. It's actually being done by hundreds of thousands, if not millions of DNA polymerases. So the DNA replication happens really, really quickly. Really quickly. Yeah. Any other things that are interesting about that, seeing this as the widest part of the circle? How often? Oh, go ahead.
Is that like wrong?
It is wrong. This is a terrible, absolutely terrible representation if you're trying to represent the amount of time.
And then also,
so this must not be representing time, otherwise I wouldn't show it to you, right? If I was going to show you a terrible figure, I would just show you a figure out of your own textbook, right? And you're like, well, why do you assign this textbook if they have terrible figures? We've already talked about this. This is free. And I could supplement it with better figures. But if I wanted to show you a terrible figure, I would draw one. And then after that, I would use one from your textbook. But no, because this isn't trying to reference the amount of time spent in each part of the cell cycle. Instead, it's trying to not necessarily the size, but the relative amount of energy dedicated to each part of the cell cycle. And the relative... just the relative amount of material that the cell dedicates to each one. And so S phase happens really quickly, but it is really, really expensive, which is one of the reasons why your cells don't just replicate like crazy, because it is a very expensive process. Another reason they don't replicate like crazy is there's really nowhere for them to go, right? It has to be a highly controlled process, because space is a valuable resource in a multicellular organism. Space is a very valuable resource. All right? Anything else you notice about the cell cycle? So the question was, what are the stages of the cell cycle and how are chromosomes involved? Well, the stages of the cell cycle are G1, which is gap 1. This is the normal functioning of the cell, right? Cell's doing what the normal cell does. You have S phase. S stands for synthesis. You are replicating the DNA. Once you do S phase, once the cell does S phase, it has to finish, or the cell needs to be set to die. Because if it has 2 copies of the genome, that's going to be really bad for the normal functioning of the cell. G2 is preparation for mitosis. So G2 is just transition, starts preparing for mitosis, starts getting things ready. The centromeres and the DNA are duplicated during S phase. And so how do chromosomes factor in? During S phase, every chromosome gets copied. And so a typical human cell has how many chromosomes? 23 pairs, 46 chromosomes, right? One from your mother, one from your father. Well, during S phase, those get duplicated. And so now instead of having 46 chromosomes, every cell actually has 92 chromosomes, if 46 times 2 is 92. It sounds right. But I don't know what ibuprofen does to your brain, but I feel like that's right. And so right here we have double the amount of DNA that the cell should have, which actually now 4 copies of the entire genome. And so G2, it's not functioning normally, it's just preparing for mitosis. In the last stage of the cell cycle, still factoring in chromosomes, the chromosomes need to be split into 2 different nuclei. Okay? They don't need to be split into entirely different cells. Notice that that's separate from mitosis. Okay, cytokinesis, when the cell actually splits, is not part of mitosis. Usually happens along with mitosis, unless you're making something special. Anyways, yeah, there are some cells in our body where you just have multiple nuclei in the same cell. Skeletal muscle cells are that way. Anyways, it's another story for another time. And so metaphase, you split those 2 copies into 2 separate nuclei. All right? So then a couple of other figures. This is just zooming in on the M phase, showing you right now what's going on with those chromosomes. How do chromosomes factor in? So in S phase, we've duplicated the entire genome. So instead of having 2 copies, you now have 4. Okay? And so now that cell has to split those into 2 separate nuclei. And so that's what happens during M phase, during mitosis. And we'll talk about that in a little bit more detail. But as you just real quickly do a rough look through these phases, here's interphase, which is everything but M phase. So interphase is G1, it's S, it's G2, it's everything but M. Okay, here you've got, normal structure to the chromosome. Here, once we get into M phase, you have like kind of our cartoon representation of a chromosome. It's cartoony, but it's actually pretty accurate based on what a chromosome looks like. But chromosomes only look like this during M phase, when they really condense down, and so that they can be split into 2 different cells. So that's what's going on during M phase, being split into 2 different nuclei, and then the 2 nuclei ending up in 2 completely different cells. All right? Any questions about that? Okay, so now we'll sum this up. So what are the stages of the cell cycle, and how do chromosomes factor in? There are 3 main events. There's interphase, which then you can divide into 3 parts. G1, where the cell grows and functions normally. S, where the DNA and the chromosome proteins and the centromeres are also duplicated there. And then G2, where the cell grows and prepares for mitosis. One of the 3 main events in the cell cycle, interphase. Another one, mitosis, where the chromosomes are segregated into 2 separate nuclei. And then the last one, what would be that last main event? Cytokinesis, where those 2 nuclei are spread into 2 separate cells, 2 daughter cells. Unless, of course, that doesn't happen, but that's rare and for a specific purpose. Let's think about this. Why would a skeletal muscle cell need to have more than a single nucleus? This is some critical thinking.
It's trapped inside its own matrix, so it can't split up, but it needs, for some reason, 2 different nuclei.
Yeah, they tend to have a single skeletal muscle cell tends to have dozens of nuclei. It's a massive cell. It's a massive cell. So almost every skeletal muscle cell runs the full length of the muscle. So your biceps runs from basically the head of your humerus down to your elbow. And so that's a really long cell. And so you need to be able to kick out mRNA that are going to make the proteins that are really viable or valuable for muscle function along its full length. Yeah.
So it does is it because it does all the things of the cell cycle minus the cytokinesis, so it just ends up being baked by no cytokinesis?
Yes. Yep, there's another way in which you can do that, get a number of nuclei in the same cell. Bless you. A little bit different. But yeah. All right. Any questions about this question? All right. So what we're going to do, we're going to take a lecture break right now. You're going to get into groups of 4 or 5 or 6, preferably no less than 4. I know I've done no less than 5 in the past, but there's like a group up in the back corner where if you do less than 5, or you don't do less than 5, you're going to have to recruit some people, but there are 4 of you, so no less than 4, preferably no more than 6. If you find yourself in a group of more than 6, I guess that's okay. Just know it's not preferable. And so what I want you to do is predict where is this cell going to invest the most in regulating the cell cycle and why. Where are you going to find the tightest levels of regulation and why? Okay, so you've got 4 basically phases to choose from. I guess you could just do this one, interphase, mitosis, and cytokinesis. But you should get a little bit more detailed than that. And predict where is the cell going to invest the most energy and the most material for regulating this process and why. Okay, take a couple of minutes, talk about it with those around you, see if you can come up with some options. Find somebody in your group that wants to share your thoughts with the group as a whole, and then we'll carry this out. Honey brown. Oh man. Oh, I know. You know, I knew before it actually happened. We do quizzes in genetics like this. He wrote at the end of his genetics quiz. By the way, I'm going to propose to my girlfriend over there.
Yeah, so I think you'd still be in G1 phase.
Where in the cell cycle is the cell going to invest the most energy and resources for regulation and why? Got about another 45 seconds or so. Wrap up your thoughts. Find somebody.
But I think it has to go through a bunch of checks before it can start to do
Oh, I love this. Some different thoughts within the same group.
I know, it's like Congress.
Gosh. I hope it's not that bad.
But my brain's not, which means nothing.
And see, but then I'm like, I was also reading, like in anaphase when the roses apart, there's a lot of regulation, making sure.
Alrighty. Again, wrap up your thoughts. Find somebody in your group that wants to share the collective thoughts of the group with the class. All right, we're going to start over here. Ladies, do you have somebody who wants to share the thoughts of the group? Hello, Maddie. You do Maddie, right? Okay. Okay. Shh. Were your thoughts during S phase or prior to S phase?
During.
Okay.
That's when like the DNA and chromosomes are being duplicated, so they need to be monitored.
Monitored for what?
For any mutation.
Yeah, right? We talked a lot about that when we talked about DNA duplication. and all the levels of control the cell has for proofreading and fixing what goes wrong. Good, I love it. We have a group back here somewhere. Is it U4? Well, we're not quite back there yet. It's right here. You have some thoughts that you want to share with the class?
Well, we also said S-phase.
Okay.
We were more talking about how, because, like you said, once it gets to S-phase, it has to go on to G2 and mitosis, or else
Yep.
the cell will die. So they have to make sure that it's what happens in S-phase is completed correctly, because they can't go backwards.
Sure.
from it.
Okay. Yes, a point of no return, right? So your thoughts are during S phase. During S phase. Okay, group in the back. Okay.
Every gene codes for a specific protein within the cell, and so while it's being copied, if something is copied wrong, and that's how you have a mutation that happens, or something gets all wacky with the
Sure.
cell cycle, which is how you get something like cancer
Right?
coming around.
Yep.
And you die.
Potentially. Depends on the type and how early you catch it. But all right, thoughts from this group in the back?
Either the G2S checkpoint or the G2M checkpoint.
Okay. So G2S or G2M. Okay. And so when you're referencing specific checkpoints, you realize something, that there's not really a checkpoint prior to S phase, right? Which means that the level of control is probably not at the cellular level, and is something else. Was this a group of 7, or did one of you split? Okay. That the whole 4, 5, 6 thing, right? Is tough. It's tough. All right, we have right here, group in the middle.
So yeah, we were kind of thinking in terms of energy, probably the S phase, but then in terms of material, we thought mitosis, because that's when it's actually splitting and all the microtubules are involved.
Okay.
And everything.
Yeah, there are certainly a lot of. I mean, the cytoskeleton always plays a big role in cellular function, right? We talked about that before. Microtubules anchor organelles in place. They provide a scaffolding for vesicles to move on, and so the cytoskeleton always plays an integral role in cell function. But when you start to look at cell division, almost everything is performed by the cytoskeleton, because that process is so highly controlled and highly regulated that virtually nothing else is happening in the cell at the time, other than ensuring that the 2 copies of the 4 copies of the genome are segregated into 2 separate nuclei. It's a very, very highly controlled process. In this group, I know there was some disagreement. So, who's representing your thoughts?
I am. So as far as energy is concerned, it invests most energy in S. It regulates the S phase probably the most, also to make sure there aren't any mutations. It also probably is going to regulate when S phase happens the most, because you don't want to get started, because then you can't go back. And the actual reason the cell exists is during the G1 phase. So you want it to do its job and then go into S phase.
Sure.
So it's going to regulate when it enters S phase. So
It's an interesting thought. It requires you to think, why does a cell exist in the first place? So if we are a multicellular organism with literally probably hundreds of trillion of our cells, of human cells, and another tens of trillions, if not hundreds of trillions of other cells, bacterial, fungal cells, part of your normal flora, an individual cell in that process, what specific role does it play? That's very interesting. It's an interesting and meaningful question to ask, because what's really interesting is if you take a cell out of its environment, almost any cell, it'll start going through mitosis. You take almost any cell in your body out of its environment, it'll start going through meiosis, because it recognizes that there's a problem. It's not a single-celled organism, and it's the only cell around. And this is really nice if you want to culture cells, because you can take a cell out of the organism and it's going to start going through mitosis. And if you can control it and keep it alive, you can keep all of those cells alive and you can build things. like hamburgers and things like that. Have we talked about this before? That in organismic biology, that's the $400,000 hamburger made by cultured beef. But it's probably the protein of the future in most of your developed countries, because trying to convince people in developed countries to eat insects is really challenging. Okay? But to try to convince people in developed countries to eat something that scientists made, they're like, sweet, right? We consume all kinds of things that scientists make. We're like, this is awesome. It must be the best thing available. Except for now, you're starting to see a trend away from that. But yeah, you can use it to regrow heart tissue. And there are a lot of researchers doing this. One of my former students is working on a project right now where they're taking hearts out of sheep. They're scraping everything off of the heart except for just the extracellular matrix. And then they're regrowing a heart from a sheep's own tissues, for the idea that you can get a heart transplant from literally anybody, and you'll take away everything cellular, just leave the extracellular matrix, which shouldn't trigger your immune response, and then you regrow a new heart with your cells that won't trigger a rejection response. It's kind of a neat idea. And again, it's because if you take a cell away, it's tended going to start going through mitosis. And so the level of control of whether a cell enters into S phase is not really regulated by the cell. The cell would love to do that. Cells are programmed in such a way that they really want to make copies of themselves, because that's what helps an organism grow and develop. And so then once that process stops, the reason it stops is typically not because the cell decides it's time to stop, but because there's no room in the extracellular matrix. It's an external control. And the way you can tell that is if you pull a cell out of its environment, it's usually going to start going through the cell cycle. Now, there are some exceptions to that. If you pull an individual neuron out, there's going to be some challenges. An individual skeletal muscle cell, because a lot of those are in that G naught, where they're just not even in the cell cycle anymore. But you can trigger them to get back into the cell cycle, which is cool because neurons don't really regenerate very well. But if somebody's got some significant neural degeneration, you can take some of their healthy neurons out, trigger them to start going through mitosis, regenerate new neurons, and then figure out a way to implant them in the correct place. But we're still trying to figure out how neurons migrate along neural pathways, because it's really weird when you watch a baby's brain develop. The neurons don't develop where they're supposed to be in the brain. They develop in one central location and then migrate to where they're supposed to be. So trying to replicate that is challenging. Yeah.
Is that because of like the hormone thing that makes them do stuff and then they?
It's because it's primarily because the level of control over moving into different parts of the cell cycle. That basically everything's default position is to keep the cells in that tissue from dividing. Mm-hmm. And then so if a cell, really what happens when a cell starts to grow out of control is it loses the ability to recognize the communication from outside. Because the communication from outside is telling it to not go through mitosis, not go through S phase, G2, and mitosis, but then it loses the ability to recognize that, or loses the ability to respond correctly and starts growing like crazy, and that's where you get tumor development. And then if it metastasizes, you've got some other things that need to shift in order for that to happen as well. So the highest levels of control are really in transitioning from S to G2 and transitioning from G2 to M, and then in the midst of M. Those are the big places where the cell actually invests a lot of energy and resources into controlling the cell cycle. Everything else tends to be externally controlled. S to G2, G2 to M, and in the middle of M. Yeah. But everything else tends to be primarily externally controlled. So now the cell is obviously investing a lot during S to make sure that happens correctly, but then there's a checkpoint at the end of S to see if everything was good, and you can move on to G2 and start preparing for mitosis.
So the reason why stomach cells are continually reproducing is because they keep dying and there's enough space just to keep doing mitosis over and over again?
Yeah. Mm-hmm. Yep, and the same reason why your skin cells keep, like, at the base of your epidermis, keep, going through mitosis like crazy, and in the roots of your hair is because things, you slough off skin cells all the time. The average mattress gains 10% of its weight every 10 years. And the primary source of that extra weight is material you're losing, which the overwhelming majority of that are dead skin cells. So in a place like this, there's a lot of dust in the air. Our dust is probably half dirt particles and half dead skin cells. But in a place that doesn't have as much, dust in the air, your dust can be, 90, 95% dead skin cells, which is cool. They're not really you anymore because they don't have a nucleus. They're just packaged, pockets of intermediate filament proteins of keratin. Yeah. All right. So here's just a picture of what's going on with the chromosomes. The chromosomes are labeled in blue, labeled in red, and green are cytoskeletal elements. And you can see the entire cell, even though the chromosomes are just really in a specific spot inside of the cell, basically the entire cytoskeleton is functioning to either maintain cell shape or to split those chromosomes apart. Very, very highly controlled. I don't think I presented the question. We just moved right into the question of where does the cell invest the most energy and resources. But the question we're addressing here is, why is regulation of the cell cycle so important? All right, so in order for organisms to grow and reproduce, cells have to reproduce. It's very unusual to have cells grow. Cells grow beyond a certain size. So as a multicellular organism grows, most of that is done by cell division. And in multicellular organisms, again, this is very highly controlled, and much of that control is beyond the level of an individual cell. It's actually tissue-wide or organ-wide control over cell division. And most of that control is suppressing cell division, because the default is just keep moving through that cycle. And so if you want to stop it, you have to suppress it. Yeah, and so the mitosis and cytokinesis function to basically take one cell and to generate two cells that are genetically identical to the parent. And so the DNA, we learned about this when we talked about DNA replication and when we talked about transcription and translation, that the DNA is basically as a composite molecule of nucleic acid and protein called the chromosome. And so if the DNA is duplicated, you also have to duplicate all of those proteins that are a component of those chromosomes as well, mainly histones and histone-like proteins. Oh, that's this point here. Now, most organisms maintain two copies of each chromosome in order to function properly. And so after S phase, what you find is that two of the four copies are actually connected together. So when the DNA is duplicated, it's duplicated in such a way that the new chromosome is physically attached to the old chromosome. Okay? So your default setting is to have two copies of chromosome 1, one from your mother, one from your father. During S phase in a cell, you duplicate both those. Your paternal chromosome is duplicated, your maternal chromosome is duplicated, and the old is physically attached to the new. And why is that? Why are they physically attached together? Any thoughts? This isn't like a true lecture break where we're going to get and start thinking in our various groups, but just thinking on your own, why does it make sense why the two copies, the old copy and the new copy, would be physically attached together? And so now each one of those chromosomes has one old strand and one new strand. That's not what we're talking about. So each of those two DNA chromosomes that each have one old and one new, they're physically attached together. Yeah.
That way, they're like a unit, so you don't accidentally. It's like easy to organize.
It is easy to organize, and keeping in mind that once you duplicate the DNA, you have to segregate it into two different nuclei. The easiest way to ensure that that happens properly is to make sure they're physically attached together so they line up together and so that they can be pulled apart. All right. The next question we're going to deal with, what are the stages of mitosis? What are the stages of mitosis? Now, some people are lumpers, some people are splitters. Okay? Lumpers like to group as many things together as possible to just try to simplify the number of steps that you have. Okay, and there's some value to that. Other people are splitters, and splitters look for clean breaks and opportunities to split things in such a way that each individual unit is easier to digest. Okay, I'm a splitter. I like to take things and to split them into different groups. And so sometimes when you go over mitosis, you'll go over 4 stages of mitosis. I like to think of 5 stages of mitosis. I just split prophase into 2 separate phases. And the text does this as well. Some people are splitters, like I'm a splitter. Other people are clumpers. And so you'll actually just have 4 stages of mitosis. So if there are 5 stages, what are they? What are the 5 stages of mitosis, if you have 5? Yeah. Prophase, prometaphase, metaphase, anaphase, telophase. Okay, so here's prophase. What's going on here? What do you see happening? There are 3 huge events that happen during prophase. And you can see all 3 of them in this diagram. Now, one of them is already complete in this diagram, but it's still one of the 3 big events.
The nucleus starts to degenerate.
Yep, the degeneration of the nucleus is one of those 3 big events that happens during prophase. Lacey?
The DNA is one becomes like.
Yeah, the chromosomes condense. Okay, that one's already completed in this picture, but that's another one of the 3 big events that happens during prophase. DNA, the chromosomes condense, the nucleus deteriorates, and what's the third? You can see it, it's about halfway there. The centromeres. Centrosomes, not centromeres. Don't call them, centromeres, completely different idea. Centrosomes, okay, made up of centrioles, but the centrosomes migrate to opposite poles. Okay, and you can see it's about halfway there. Because these are duplicated during S phase as well, and so they need to migrate to opposite ends of the cell. Because that's where the microtubules grow out of, is those centrosomes. And so those are the 3 big events during prophase. Chromosomes condense, the nucleus deteriorates, the centrosomes migrate to opposite ends of the cell. Now prophase is done. Prometaphase. There's one big event that happens during prometaphase. And you're like, well, why don't you just lump it with prophase then? Because it's a nice clean break. It doesn't happen simultaneously with the other 3. This picture is a little bit misleading, because it makes it look like chromosomes condense, then nucleus deteriorates, and the centrosomes migrate. But all 3 of those are happening simultaneously. As the chromosomes are condensing, the nucleus deteriorates, the centrosomes migrate to opposite poles. There's a very clean break when you get to prometaphase. And there's only one main event here, and what is that? Yeah. Yeah, the microtubules connect with the chromosome at what's called the kinetochore. Okay, so during prometaphase, your microtubules make physical contact with your chromosomes. And now once the microtubules are attached to these chromosomes, what can you do? You can separate them and you can move them wherever you want them. Because remember, things move inside of a cell really only as they are walked along a microtubule. Now there's exceptions to that. Really small materials are going to diffuse around a cell. But for the most part, anything big, like a chromosome, can only move along a cell as it's walked along a microtubule. Okay, but once those microtubules have made contact, now you can do that. And it's actually not the microtubules attached to it, just like it's not the microtubule attached to a vesicle. It's a motor protein attached to the chromosome, which is also attached. So the motor protein is also attached to the microtubule and to the chromosome. The kinetochore, and it's in the next slide.
So motor proteins connect to the
Microtubules and to the chromosome.
and that's what causes them to split eventually as the motor protein starts walking up the microtubule.
You would think but the actual what causes them to separate is the microtubules start shortening.
Oh.
Yeah, the motor protein walks them to the end of the microtubule, but motor proteins can only walk in one direction. So it'll walk it to the end of the microtubule, which will line it up in the middle of the cell, and then in order to split them, you have to start taking apart the microtubules.
Okay.
And you would think that you should take them apart at the very end. This is way more than you need to know. This is in more detail than what you need to know. You'll learn it in genetics. But it actually takes them apart right next to the chromosome and not at the very end, next to the centrosome. It's kind of strange why that happens. I don't know. All right, so here's the kinetochore. It's an actual protein complex that attaches to the chromosomes, and then the motor protein attaches to that, and the motor protein also attaches to the microtubule. Okay, but that's what happens during prometaphase. Yeah, you actually get a physical attachment between the chromosome and the microtubule. Not directly, indirectly. There's more involved with that, but you physically attach to those chromosomes, and now you can move them wherever you want them. You line them up in the middle of the cell, and you can split them apart when it's time. All right. Real quick, we'll do this, and then we'll be done. During metaphase, there's also one key event that happens during metaphase, and that's your chromosomes get to the end of the microtubule. And when they find themselves at the end of the microtubule, they're lined up in the middle of the cell. It's called either the metaphase plate or the metaphase line or the equator. A lot of different names for the same thing. Usually, my favorite is the metaphase plate, because then that makes you think metaphase, and then, metaphase literally means middle, and it's not referring to the middle of the state of the cell, it's referring to the middle of the process. Anyways, so there's one key part of metaphase, and that's the chromosomes line up in the middle of the cell. Okay, and that's because they got to the end of the microtubules that they're attached to. All right, we're done. It's 12:40, we're out of time. Have a wonderful conclusion to your Monday.